First of all, we will discuss about the apoptosis, their function, as well as first of all, we will learn about the introduction of the apoptosis. So here is the person which that is contain a cell, somatic cell, which that is able to divide and maintain. It means the division of the cell will be maintained throughout the young age, while the decrease during the old age. So here is the mitosis in healthy cell will be occur. While in the case of injury of the cell or inflammation or other stress creation will lead to enter into the apoptotic pathway. But important thing is that here is the injured cell, the injury to cell, which that is genetic error remains, but it can be all errors repairs, and in this way the repairs is basically can be occur, and in this way the repairing of the cell will lead to again go into the mitosis in healthy cell. So that is the healthy cell and damaged cell. So it's contain DNA, genetic material. So their genetic error will remain. If so, it can be all errors repairs after the reparation. So the mitosis in healthy cell will again like that. But in the case of if the genetic error is more and some errors remain and it is not unable to repair, so the additional injury to cell hyper injury. So this hyper injury is the some errors remain, and these some errors will not cell unable to repair errors. After this cell will not able to repair, so it will go into the cell apoptosis rather than go into the cell division, because the cell division if continue, so the cancer causing gene will be activate, the oncogene, and the proliferation of the cell and uncontrolled cell division will be occur. That is very dangerous, but the cell program itself to die via a apoptosis. So let's begin to understand the control of apoptosis. So the apoptosis is complication and tightly controlled process. In this way, that is very important for the controlling of apoptosis, because without this, the cancer-causing gene will be activated due to the mutation and damage of the tissue. Damage of the DNA. So the regulation occurs through positive signal. Positive signal means the ligand, which that is the death receptor, will bind with the positive signal. After the positive signal bind and required for cell survival, so the growth factor, for example, such as insulin, somatostatin, mTOR pathway, which that is the positive signal for the proliferation of the cell. But the when it's unable to balance the cell, so the one of two things may occur: the rate of apoptosis too high, such as HIV infected helper T cell, induce apoptosis in neighboring T cells and limiting the immune response to the virus. So the rate of apoptosis becomes too low, such as low rate of apoptosis becomes too low. Example is a low rate of lymphocytes apoptosis. So the lymphocytes apoptosis is associated with and other active the immune system. So in this way, the another things you should need to understand that is the apoptosis is the death of the cell. So if the insulin and growth factor will be bind with the receptor to proliferate, so the growth of the cell will be occur more. If the damaged cell, so when bind with the growth factor, so after the binding with the growth factor, the cell proliferation will be more, and the uncontrolled cell division due to the oncogene. So important thing is that let's function we will discuss here. The just is the apoptosis is an important evolutionary adaptation because it is the multicellular property, multicellular organism which that is used for the apoptosis, and because it allows organism to destroy their own cell. So the well, perhaps if that part of cell has become dangerous to the rest and damaged cell can become cancerous cell. So the cancerous cell prevention is the best mechanism is known as apoptosis. And the next is little bit you should need to understand about that is the autophagy. So the autophagy is the also part of the apoptosis, but the self-eating of the part of the cell is also the part of apoptosis in the mesosome. So the mesosome is very important for the apoptosis, but the apoptosis is the little bit part. For example, the mitochondria damage. So here is the apoptosis can be micro, macro, or mitochondria, which that will engulfed by the mesosome. So, but in this case, the whole cell cannot destroy. mTOR pathway is very crucial to stop it, and starvation stop the mTOR pathway. If the stop of the mTOR pathway will lead to cancer proliferation, the cancer cell proliferation. Why? Because the mTOR will be inhibited by the starvation, though, so the fasting is very important. So apoptosis pathway we will discuss here is the intrinsic pathway and extrinsic pathway. Intrinsic mean the intrinsic path to cell death by the stress and damage the cell inside, not outside signal, while the extrinsic is the signals is received from outside the cell in instincting to it commit the programmed death. So the programmed death through a extrinsic ligand. So here is the cell I am drawing. So let's begin to understand about the damaged cell. So it's contain a plasma membrane. This plasma membrane is made up of phospholipid of bilayer, while the extrinsic pathway is the extracellular space are present, where the extrinsic ligand are present. This extrinsic ligand will bind with the extracellular domain of the receptor is known as the death receptor. Is made up of trial and FAS, FAS and TRAIL, trail and FAS. And this is the important things. The intracellular domain is also attached with the FAD, so the FADD. So FAD is bound with the procaspase 8 and procaspase 10. This procaspase 8 and procaspase 10 is the inactive, while after the binding with the ligand to stimulate, after the stimulation to become a mature this caspase, procaspase to cleaved their segments after the cleavage formation to produce the caspase 8 and caspase 10. This caspase 8 and caspase 10 is dispersed in the cytoplasm for the apoptosis program. So in this way, the cleavage will be a curve of the bead protein. This bead protein is very important and crucial mechanism for the apoptosis. So the bead is the inactive, and if the it is inactive, if so let's begin to understand. Here the caspase 8 and caspase 10 is the phosphorylation of the caspase 3 and caspase 7. So this phosphorylation will be occur throughout this pathway, and this caspase 3 and caspase 7 is also important for the apoptosis. So remember about that. Let's begin to understand. This was the extrinsic pathway, while the intrinsic pathway we will discuss here. Just first of all, we will need to understand the caspase 8 and 10 will cut cleavage formation through enzymes, the cutting of enzymes to bead to convert the T bead. So the small T bead, and this small T bead is the protein as the enzymatic protein, which that will activate the bags and bag. This activated the bags and bag are present in the cytoplasm. So in this way, important things that here is the MCL1, BCL2 and BCLX, which that is a part of protein. This protein will inhibit this reaction to convert the T bit, and in this way here is the BH3 only protein. This BH3 only protein is also inhibit, and in this way inhibition of this protein will not lead to apoptosis. So this was the intrinsic pathway. Intrinsic means it when the stress and radiation damage the DNA, etc., will stimulate the BH3 only protein, which that is the pro-anti in apoptosis protein. So this pro will become mature to activate the bags and back. So this is the two pathway. This is the internal pathway, is the intrinsic, sorry. So extrinsic and intrinsic pathway also need the backs and back collectively. So in this way, the activated the backs and back is together is a simple and same mechanism. So the intrinsic, the backs and back active to activate the MOMP, the mitochondrial outer membrane permeability. That means, and in this way, the here is the cytochrome are present in the inner mitochondrial membrane, and that will efflux of these things through a MOMP, and the backs and back is active to stimulate this and to produce the outflux of the SMAC, as well as other is the cytochrome C. The cytochrome C is the signaling molecule, and SMAC is also the signaling molecule. Will inhibit the XIAP, and this after the inhibition, this inhibition will lead to inhibit the procaspase 9 to convert the caspase 9. So that will inhibit during these things. But important thing is that during the apoptosis, that should need to not inhibit. So that means the XIAP will SMAC will inhibit. So it means it will not inhibit the procaspase 9 to convert the caspase 9. And in this way, here is the APAF1 oligomerization will be occur. And in this way, the APAF1 as well as the cytochrome C. APAF1 and cytochrome C will bind together to form a minus sounding apoptosome. And this apoptosome, which that is convert the procaspase 9 to convert the caspase 9. After this caspase 9 production, this procaspase 3 and 7 again, you should need to understand, remember about that previous is the extrinsic pathway was the same. But here the procaspase 9, 3 and 7 will activate through caspase 9. And caspase 3, and here you can see this is the mechanism. This is the intrinsic pathway which that require the outer ligand, while here is the damaged DNA which stimulate the MCL1, BCL2 and BCCLX, while inhibit during the healthy cell. While here is the caspase 3 and 7 activate to break down cellular material. So remember, this is the major mechanism you should need to understand. I hope you are getting this concept about the apoptosis. So here is the inhibitor. What is the inhibitor of apoptosis? Here is the growth factor. For example, insulin. For example, the growth hormone or other things which that is a growth factor will bind with a growth factor receptor after the tyrosine kinase receptor. This is and will lead to activate the cancer progression by the inhibition of the apoptosis, while also inhibit the ligand binding the death receptor. So thanks for watching. Bye.